Hi everybody, I'm Dr. Biomed. Welcome to the Biomed Cafe channel. In this channel, complex biological and medical contents are presented in a simple, classified, and summarized form. Please subscribe to this channel, and if you like the contents of this video, please like it. The topic of this video is the structure of proteins in cell membranes. Let's go to the content. First of all, I recommend you to watch one of my videos. Its name is The Classification of Cell Membrane Components. Okay, now I want to speak about proteins in the cell membrane. Proteins in the cell membrane are in two groups. The first group is integral proteins that are tightly attached to the membrane. Transmembrane proteins are a type of integral protein. They span the cell membrane in both the intracellular and extracellular environments. They can be divided into three subgroups. One of the transmembrane protein subgroups is the single alpha helix. It spans the lipid bilayer only once. It has three domains in its structure: the extracellular domain, that its hydrophilic segment and the largest domain, the transmembrane domain, that is the smallest domain, around 25 amino acids, and it's inserted into the membrane bilayer, and the intracellular domain, that is the hydrophilic segment. It's classified topographically by topic. It's located in the inner membranes and sometimes outer membranes of bacterial cells, and in the plasma membrane of eukaryotic cells. It has role as regulatory subunits and as structure stabilizing subunits in large multi-protein transmembrane complex such as photosystems or the respiratory chain. Multiple alpha helix or multipass membrane protein is transmembrane protein that spans across the membrane more than once. It has three domains in its structure: the extramembrane domain or ECD, the transmembrane domain, and the intracellular domain or ICD. It's classified topographically polytopic. It's located in the inner membrane and sometimes outer membrane of bacterial cells, and in the plasma membrane of eukaryotic cells. It plays a role as transporters, channels, receptors, membrane anchoring domains of enzymes, and proteins responsible for cell adhesion. A rolled beta sheet composed of tandem repeats that twist and coil to form a closed structure in which the first strand is bonded to the last strand by a hydrogen bond. The number of beta strands in rolled beta sheets varies widely from as few as 8 strands to as many as 22. It has three domains in its structure: the extracellular domain, the transmembrane domain, and the intracellular domain. It's classified topographically polytopic. It's located in outer membrane of gram-negative bacteria, in outer membrane of mitochondria, and in outer membrane of chloroplasts. It plays a role as ion transporters and translocases. Non-transmembrane proteins are another type of integral protein. They don't span the cell membrane. They can be classified into two subgroups. Proteins with an amphipathic alpha helix interaction. This non-transmembrane protein is anchored to the cytosolic surface by an amphipathic alpha helix. It has two domains in its structure, the intracellular domain and the intramembrane domain, that partitions into the cytosolic monolayer of the lipid bilayer. Its interaction is amphipathic, is classified as topographically monotopic, is located in the membranes of bacterial cells and in the plasma membrane of eukaryotic cells. It has a role in metabolism, in biosynthesis, and in cellular signaling. Proteins with a covalently bound interaction. These non-transmembrane proteins is attached to the bilayer only by a covalently bound lipid chain in a cytosolic monolayer. It has two domains in its structure, the intracellular domain and the intramembrane domain. Its interaction is covalently bound, is classified as topographically monotopic, is located in the membranes of bacterial cells and the plasma membranes of eukaryotic cells. It plays a role in metabolism, in biosynthesis, and in cellular signaling. Peripheral proteins interact with the surface of the lipid bilayer of the cell membrane. They do not enter into the hydrophobic space within the cell membrane. One of the peripheral proteins is lipid-linked proteins. They do not have a hydrophobic region of amino acids. They include amino acid sequences that enable them to bond to the phosphate heads of lipid molecule through two interactions, hydrophobic loop interaction and ionic or electrostatic interaction. They can bind to the membrane without being locked to it. They are located in the cytoplasmic membrane of bacteria, in the membrane of mitochondria, and in the plasma membrane of eukaryotic cells. The main role of them is in maintaining the intracellular cytoskeleton, in maintaining components of the extracellular matrix, and in transferring a small molecule or electrons. Another peripheral protein is a protein-linked protein. Like the lipid-linked protein, the protein-linked proteins do not have a hydrophobic region of amino acids. They include amino acid sequences that enable them to bind to the integral proteins through non-covalent interaction. They can bind to the membrane without being locked to it. They are located in the cytoplasmic membrane of bacteria, in the membranes of mitochondria, and in the plasma membrane of eukaryotic cell. Their main role of them is in maintaining the intracellular cytoskeleton, in maintaining components of the extracellular matrix, in transferring small molecule or electrons, and the activation of other proteins and enzymes. Thank you for watching. Please give me a comment and tell me what topics you would like to be presented in this cafe. The next video, I will talk about the structure of carbohydrates in the cell membrane in a simple, classified, and summarized form. Until the next video, bye!